Okay, before we pick up where we left off, just a couple of brief announcements. First off, apologize, these videos keep going up a little bit late. I don't know about y'all, but boy, this online stuff is killing me, right? It seems like it's a lot more work maybe than the typical uh, lecture prep and lecture, but we'll get through it. Hopefully you're getting through it okay. You've settled into a pattern. I will post for you an assignment. I'll probably just email it out to you, or maybe I'll post it on Blackboard. I'll let you know when it's available in either case, just like we did for the last one, right? I think I put it on Joule. That seemed to work. So it'll be 10 questions. It'll be due on Monday. It'll go up on Friday. Okay, it'll cover content from this week. Also on Monday will be the exam. I'm working with Sister Christine to put the exam, to embed it in Joule, such that you'll have access for a limited amount of time, probably 60 minutes, and you won't be able to close the page and open it back up, right? So once you start it, you have to sit down and finish it. I've worked through different options with respect to how to proctor it. I just don't think that's going to happen. I would ask that you do this honestly and without using your notes or your text, right? I think that there are folks in this class who are going to do it honestly. Certainly it's not fair to anyone, to yourselves, to them, to me, if you don't do it that way. So I would just ask and expect that you do it honestly. The plan for today is to finish chapter 7. That's what the exam will end on, right? So chapter 5, 6, and 7, which is about the same amount of content page-wise in the textbook as the first exam. If I do get through that, I might add a couple of extra videos tomorrow to get through the content that I'd like to get through. But what I would say is the exam will be limited to what we cover in those chapters. All right, so we'll pick it up right away. We were talking about different types of kinases, and we left off with MAP kinases. MAP kinases are unique because, many protein kinases are activated by second messengers. Well, sometimes kinases are activated by other kinases, and that's what MAP kinase is. So mitogen activated protein kinase is what that stands for, mitogen, because it was discovered in cell cycles. The MAP kinase is activated by other kinases. And there are other families of kinases that are like this. ERK kinase is another one. So MAP and ERK are similar families in that they're activated by other kinases. And this is how the process works, right? Here you see MAP kinase, and it's inactive. And it only becomes active when a stimuli activates it through a receptor that's on the cell surface, okay? And this now activates MAP kinase. It is able to phosphorylate, as kinases do, MAP kinase. And now MAP kinase is active. Its target is another kinase, MAP kinase. And it can phosphorylate MAP kinase and cause it to become active, okay? So there's this sort of cascade of kinases and cascades of phosphorylation events that finally allow MAP kinase to become active, and it's shown here changing something at the level of transcription. So we will talk about that type of thing next, but obviously this is, it feels a bit unusual. Kinase to kinase, okay? The goal is, or the underlying theme here is one kinase activates another, until the final one, right, MAP kinase, has some effect. So within neurons, this pathway typically gets activated by things that aren't neurotransmitters necessarily, but you see growth factors or cytokines, various signaling molecules that aren't neurotransmitters, okay? And so that's the upstream stimulus. The downstream effect is, like I mentioned here, they're often transcription factors, okay? So activated by various signaling molecules that aren't neurotransmitters, right? Growth factors. Cytokines. All right, so basically these activators are not released by other neurons, right? There's not a presynaptic and a postsynaptic side, though these receptors for these growth factors, cytokines, etc., do exist on neurons, okay? And after this intracellular cascade takes place from one kinase to the next, the result are changes oftentimes in transcription. So the MAP kinase cascade is often changed to a change in protein production by regulating transcription. Before we get into the next portion of the chapter, which is on nuclear signaling, I do just want to mention phosphatases, protein phosphatases. Right, if you recall, protein phosphatases are proteins that dephosphorylate, right? They tend to shut off processes. The three of them that are pretty well characterized: PP1, protein phosphatase 1, 2A, as well as 2B. Okay, they're generally less specific than kinases, right? We talked about kinases having one particular target. Protein phosphatases can dephosphorylate many proteins, relatively non-specifically. So they're not as specific as kinases with respect to their targets. We'll talk about maybe one which is sort of unique. 2B, right, protein phosphatase 2B, needs to be activated, right? So these things aren't always on. They have regulatory subunits as well. And 2B is actually activated by calcium calmodulin complex. All right, it binds to its catalytic subunit, which displaces the regulatory subunit. So again, there's some autoinhibition, and 2B, when CAM is around, will be active because the CAM will displace the regulatory subunit. Maybe we should write this down. Displaces the regulatory subunit, right, which is typically bound catalytic subunit. All right. So this is pretty strange, right? Because protein phosphatase 2B and CAM kinase 2 are activated by the same calcium calmodulin complex, right? Now, typically they have different targets, but it's interesting, I think, to think about, okay, we're going to activate a kinase, which turns things on using calcium calmodulin, and we're going to activate a phosphatase, which turns things off by using calcium calmodulin. It's probably atypical, right? Not the way that I would have necessarily thought it would be designed. All right, let's talk about nuclear receptors now. And before that, just a real quick overview of how nuclear transcription factors work, right? Because a lot of what we're going to talk about work as transcription factors. So this picture didn't show up very well, but the idea is, right, that in order to activate transcription, mRNA has to be made, right? And so we would call this transcription review. Right, and so mRNA, right, must be generated from DNA. You know this. And so the first thing that has to happen is chromosomes decondense, right? They need to be looser. And once they're loose, different proteins can bind, right? Transcription factors can bind. right? And so a particular sequences of DNA, right? Transcription factors, which are proteins, bind, and what they do is they tend to help recruit or stabilize RNA polymerase. Right, so this is a really broad overview, but so these transcription factors, I'll abbreviate them TF, right, can help recruit or stabilize or bind the RNA polymerase. And it's, of course, the RNA polymerase enzyme which produces mRNA, and this mRNA eventually gets translated into a protein, right? And so that's how we connect this process of transcription to translation and protein production. Now, oftentimes, genes are regulated by whether or not transcription factors are present, okay? And transcription factors can be either active or inactive, right? And so inactive transcription factors can become active. How? Maybe it's a second messenger. Maybe they're phosphorylated and now they become active. Maybe calcium concentration goes up and that causes transcription factors to become active. They bind particular areas of the DNA that allow mRNA signaling or activity, I should say, to take off. Okay, so what we're going to look at now is how certain genes are regulated by transcription factors that rely on nuclear signaling. Okay, signals that are going to the nucleus. So let's first look at how CREB is regulated. CREB is a transcription factor that is found ubiquitously, right? It's found in all cells, including neurons, okay? So the first example of nuclear signaling that we're going to look at is via CREB. So maybe we can call this nuclear signaling in neurons. Can be our title, as we're going to talk about 3 different pathways here, and the first is via CREB. CREB stands for cyclic AMP response element binding protein. So cyclic AMP response element binding protein. So there are some hints in the name, right? Cyclic AMP is one, and the fact that it's a protein is a second one. It is a transcription factor, okay? And so typically it's bound to DNA. But generally, right, when it's bound to DNA, as long as it's not phosphorylated, it's not very active, okay? So once CREB is phosphorylated, right, so CREB that's been phosphorylated, this gives us lots of transcription. Whereas regular CREB that doesn't have the phosphate attached to it, very, very limited transcription. So we want to look at how CREB is phosphorylated by signals that we've talked about so far to induce a transcription. So we'll start that on the next video.